Well, I would have liked to see a lot of you in person, but I guess also the Zoom is fun. So I will present to you the next 40 minutes or so a little bit about what we are unintentionally learning, and we are realizing that this is a very important role of repressors in general in stem cells and pluripotency. And that's why the title of my talk is about liberating mammalian pluripotency from repression. So in general, my lab studies early development. And the big question is really, there's so much drama happening after fertilization and formation of the blastocyst and then implantation. We have a lot of events that are happening that we would like to know: specification of trophoblasts, the inner cell mass, DNA methylation goes down and then up, X chromosome in females gets reactivated and activated. And it's very hard to study these in vivo because they happen so fast and we have very limited material. So many times we take in vitro stem cells as a tool to study this. And I want to also ask, of course, we're trying, although I won't talk about it a lot today, but also we're trying to compare mouse versus human because there's a lot of knowledge that is missing in human stem cells. And when we talk about pluripotency, we should remind ourselves that unlike adult stem cells that self-renew and exist in our body, pluripotent stem cells in early development is a transient state. They don't really self-renew. They exist for about 1 or 2 days. But if we think of the stemness in the inner cell mass as a synthesis, some kind of a climax, we should look at, there's the assembly of it, which is the first vector, and then how it is also resolved, how the exit happens. And also we're learning that this happens gradually and it is very carefully controlled. And so in vivo, we're talking about assembly and resolution. In vitro, we're also talking about maintenance because we can artificially keep these cells in a very useful way in a dish. But I want to emphasize that this is actually an in vitro artifact. So pluripotency is transient and dynamic in vivo state. And we can keep these cells by giving them the right cytokines. For example, if we know LIFSTAT simulation, Wnt promotes pluripotency, inhibition of ERK promotes pluripotency, and we talk about this. And by giving the right cocktail, we can preserve these cells indefinitely and compare them to what's happening in vivo. So as well, we've been learning over the last 10 years that stem cells can come with different flavors. So just as I told you, in vivo, it can change from pre-implantation to post-implantation. We can get different snapshot of stemness of early and late stem cells, or what we call them more naive stem cells, the early ones, and primed stem cells, the more advanced ones, by giving different conditions. For example, the naive ES cells that are more similar to inner cell mass depend on LIF stimulation and inhibition of ERK, versus the more advanced cells that, again, they are still stem cells, they're pluripotent cells, prime stem cells, depend on FGF and activin. And there are differences. For example, the primed cells are more flat. They express certain genes like OCT4, just like the naive, but they're not fully differentiated, but they started differentiation. For example, X chromosome is already inactive. DNA methylation is already high in them. So they're somehow primed to the initiation of differentiation. And this is where we start. We know a lot about ES cells in the early stages, but we have limited knowledge about how the change from naive to primed happens and how primed cells are maintained. And this work 4 years ago was important for us. We did this with Gidi Rechavi, and we just wanted to ask what is regulating the transition between naive and primed cells. And the first thing we did is simple, just to take ES cells that are green in the stem cell states, whether naive or primed, grow them, and knock down 40 epigenetic regulators and ask, can we find ones that are important only for the primed state? So you could see here in the bottom, what I highlighted in gray, we found many genes and that if we knock them down, the naive cells, the GFP in the red is still about 100%. The cells do not differentiate. But in the blue, the primed cells, the GFP goes down, meaning they start differentiation. So there are genes that are only important for the prime. And if you look at them, we have dicer, which is microRNA, DNMT1, which is the DNA methyltransferase, EED, and SUS12 are from polycomb. So these are all repressor complexes. And this is already hinting that somehow the primed cells need epigenetic repression. And if you remove these repressors, the cells deteriorate. And we'll get back to that. And one of the genes also we looked at is METTL3, which is an RNA methyltransferase. And that's why we joined with Guido Rechavi. And this is also a repressor. So to dig deep, what is the RNA methylation doing? We did a lot of analysis. And I'll show you the summary results. What we found that RNA methylation is important for destabilizes RNA transcripts. So if a transcript is methylated, it is destabilized. So when you take on the left side the naive cells, the pluripotency genes like NANOG and SOX2 are methylated. So if you remove methylation, these transcripts are now stable. They don't get degraded. So the pluripotent cells, we call them, become hyper-naive. They are very strong. They're actually more stable. If you go to the right, you have the primed stem cells. The cells start to express lineage commitment markers like Gatad2a, SOX17, which are methylated. So if you remove methylation at the primed state, now the differentiation genes are becoming heavier, and the cells tip towards differentiation. So that is why, first of all, the primed cells, if you remove RNA methylation, they differentiate. So if we zoom out of the primed pluripotent cells, this finding was very important for us because it showed for the first time that the same pathway, in this case RNA methylation, has completely opposite effect on naive versus primed. Naive cells are actually happier without it versus the primed cells deteriorate. So there's a very strong difference between them. And second of all, we noted that a lot of these repressors are important for the primed cells and not the naive. So this led us, first of all, a lot of conclusion that the flavors of stem cells that are different can have really opposing effects of certain genes. So if we want to look at gene X, you really need to define in your paper which condition you are working with. And there are papers in literature seemingly contradicting each other's, for example, on the REST complex, and one saying it is good for pluripotency and one saying it's bad, but actually all these papers are right. It's just that the people have been using a little bit more naive or a little bit more prime conditions, and that can have very different outcomes. So we have to be very careful about this. Second of all, that if we want to talk about this naive pluripotency in the inner cell mass, it seems that it is a unique state that it doesn't need epigenetic repressors. So somatic cells, as you know, we need microRNAs, we need DNA methylation, but these really early cells they don't need it to sustain themselves. They can do fine without it. They need it to differentiate and become primed. And this actually brings us to asking about human stem cells. And if you search the literature for human knockouts for any of these enzymes that I mentioned, they don't exist so far. Everybody who's tried to do this, you'll delete one allele and the other one stays in frame. And the reason for that most human stem cells that are used today are actually more primed. And I will not talk about this today, but actually we are using this quality for the ability of stem cells to live without these inhibitors to define how human naive cells should look like. So this is a little bit first part about repressors and stem cells. And then we're starting to talk about not only how to sustain stem cells, but how do we reprogram them to take a fibroblast and turn it back into a stem cell by the Yamanaka approach. So as you know, the Yamanaka approach works very well. However, the efficiency is low. It can be up to 1, 5, 10%. Efficient of the cells becoming iPS cells after they see the 4 Yamanaka factors. And the process is not only not efficient, but it's asynchronous, meaning let's say you take 100 cells, it doesn't matter what they are, you cannot predict a priori when and which one of them will convert. So this is what we call as random or stochastic reprogramming. If you look at different reprogramming, for example, like nuclear cloning or in development, the stem cell markers like OCT4 always come at the same time, within 1 to 2 cell division. So basically, if you plot a graph, you will see that the progression is a stepwise. So it's not really random. You can predict a priori. And this is very different between the processes. And one big effort in the field was really, can we change the Yamanaka approach to become deterministic, meaning nearly all the cells convert together at the same time? And if you actually, again, search literature, people have been successful to increase the efficiency from 1% to 10% to 20% by, for example, putting more activator, more stem cell genes, and also putting more inhibitors and so forth. But the glass ceiling, let's say, of 20% efficiency hasn't been shattered. So that even the different factors are not adding up. So we thought a bit differently. Maybe it's not about adding more activators, but maybe that this reversion has a repressive element. Maybe there are internal repressors that are preventing the cells to go back. And so the question was, maybe we can find one or two repressors that if we remove them, we remove the inhibitors, maybe the efficiency now will start becoming very, very high. And indeed, we stumbled upon this complex that has been around for 20 years, I would say. It's very important, but also it's been poorly studied and we don't know about it. And also there are very little good reagents, but we follow that path. And it's a complex called Mbd3 NuRD complex. It's a repressor complex. It's a histone deacetylase complex. And Mbd3 is a gene of a family of DNA methyl binding domain containing. And you have 1, 2, and 4. And actually Mbd3 is a little bit the weird son of the family. It is the only one that actually does not bind DNA. It is the only one that its knockout is embryonically lethal. And the cells, the mice die after implantation. And if you take ES cells and knock out Mbd3, the ES cells become very stable, or what we term as hyper-naive. And what was shown by others, that Mbd3 in ES cells represses the pluripotency genes. So if you remove it, the pluripotency genes are expressed at higher levels. So we wanted to dig deeply more on this complex and to revisit the literature. So there were papers claiming that without Mbd3 in the embryo, one cannot derive ES cells. However, if you look in these papers, if you look at the inner cell mass, you can see OCT4 positive cells in the inner cell mass of these embryos. So that was weird. And we tried again to derive ES cells, and indeed we can derive them. So ES cells without Mbd3 are sustained in vitro and in vivo. Lethality, as I said, it happens at the primed state, post-implantation. So you cannot make iPSCs, but ES cells, you can make them. Now, the other thing which with Mbd3 happens, it is if you completely remove the protein in somatic cells, the cells will stop proliferating. And for iPS reprogramming, you must have proliferation to become iPS cells. So to deal with this, we actually were lucky to track mutants that are flux minus alleles, as you can see here. So these mutants have still 20% residual protein in them. So if we take fibroblasts from these flux minus cells and reprogram them, we were very stunned to find that we can get up to 100% efficiency in reprogramming within 8 days. If you can look here, we plate at the single cells and look after 8 days, and we're getting all the cells, and all the cells within a colony become iPS cells. And that was very dramatic. We haven't seen this with any mutant we've dealt with. And this was published back then. And as a mechanism, we wanted to understand what is happening. What we found that the Yamanaka factors, these OCT4, SOX2, KLF4, and make OSKM, when you induce them in somatic cells, they bind Mbd3, which is expressed on all our somatic cells, and go to the pluripotency genes. So basically, you can see here by ChIP-seq analysis that the Yamanaka factors bind the pluripotency genes here in blue only after they are induced. So basically, we're trying to express the pluripotency genes, but they bind repressor that is found there, which makes a negative effect. So we made this analogy to basically to the gas and brakes mechanisms, or driving a car by stepping at the gas and the brakes at the first same time, which is not a good thing. So what's happening when we express the Yamanaka factors, they bring a lot of activator complexes like WDR5, and they activate the pluripotency genes. But because Mbd3 NuRD is also found, they actually bind it and they bring it to the same loci, and that creates a clash to activate the pluripotency genes. But if we remove the NuRD complex, then basically pluripotency proceeds very smoothly. So as I mentioned to you, this is like the car. There is the gas pedal, which is the green on the left here, but also there is the brakes pedal. And if you step at both of them at the same time, the car most of the time doesn't drive and spin around. So when you need to have this coordinated, and the repressors need to be neutralized at the right time and at the right level. And this is where we ended there. And since then, we wanted to understand a lot more about this. We wanted to understand what is this Mbd3 NuRD complex, what isoforms that are relevant. Also at the technical level, we were a little bit unhappy about having to work with very specific mutants that are hypomorphic and not complete null. As you can imagine, getting the right level of depletion can be annoying and technically hard. So two very talented students, Nofar Moore and Yuach Rice, asked, okay, maybe we can find another component of NuRD complex that if we deplete it at 100% levels, we don't get this block in cell proliferation, which we don't like, but still it also disrupts NuRD from functioning early in the process. So what they did, they said, okay, let's take all known knockouts, all known components of this big complex and knock them down one by one and see which ones boost iPS cells and which ones do not block proliferation. And this is what you can see here. We did the knockdown for the different components and looked at proliferation. You can see here, as I told you, Mbd3 knockdown, and here we completely ablate the protein, proliferation goes down. But they noticed a component called Gatad2a that if you completely deplete it, proliferation is not affected in fibroblasts and in somatic cells. This was promising. So they went then to look at iPS quantity, and they do the reprogramming, and they look at the number of colonies that are stained by alkaline phosphatase. And you can see here, similar to the Mbd3 knockdown, you can get a very strong increase by knocking down Gatad2a. By the way, not its homologue Gatad2b, which is only a very poorly studied protein. So what is Gatad2a? Again, this is only 7 papers on PubMed, and actually 8 because we published since then. It is also known as P66 alpha, and there are very little papers about it, but they are important papers. One paper did the knockout for these embryos, and they also die exactly at the same time where Mbd3 knockouts die, at 4.5 after implantation. So to prove, also to look at iPS efficiency, what they did, they took different iPS lines, and we insert the knock-in OCT4 GFP reporter, which is a very specific reporter for pluripotency. Then we knock out by CRISPR Gatad2a. We take the isogenic wild type and knock out ES cells or iPS cells, make a mouse out of them, derive cells, whether it's fibroblasts and B cells, and we give them doxycycline to induce the Yamanaka factors, these OSKM, and watch reprogramming efficiency. And as you can see here, in the Gatad2a knockout versus the isogenic wild type, in 6 days already, you can see a very dramatic increase in efficiency. And if we do this by single cells, you can see here again, just like the Mbd3 hypomorphs, in the Gatad2a complete knockouts, the efficiency goes up to 100% in 8 days. To show you that this is, we're not biasing this, we're not selecting, we did this also at the bulk population. We plate the cells and we fax them without passaging every 8 days, every day within 8 days. You can see here in the wild type, and we're looking at the OCT4 GFP, how in the wild type in the 8 days, the efficiency about 12%. And you can see here on the knockout how the entire population slowly goes up to 96.5 of becoming OCT4 GFP knock-in iPS cells. We can also did gene arrays, and you can see that how the wild type cells after 8 days of dox do not become iPS cells because, again, most of the cells are not reprogrammed, versus here in the knockout, you can see how the vector with each day, they become eventually identical to the ES cell signature. And if we look at the gene expression, you can here see how the MBD3 low cells, every day the signature slowly becomes completely like iPS cells, ES cells. The wild type cells, again, if you take the bulk population, they do not have the ES cell signature because the iPS cells are very minority in them. And again, the Gatad2a knockout, basically you get an identical signature like the MBD3 low. Here, each line is the same gene. And again, you can see that the signature are nearly identical. So actually, I want to skip this part. What's really also has been interesting for us is that we wanted to look at Gatad2a versus Gatad2b, because Gatad2b is very similar to Gatad2a, but also only has a difference in the C terminus, because the A has this kind of free self-organizing domain in it. And again, here you can take the knockout of Gatad2a, and the efficiency is very high. If we put Gatad2a transgene knock-in, you can see the efficiency goes down again. But Gatad2b does not block the reprogramming. And we did this also by the knockout by Gatad2b. So we're really talking about Gatad2a and not Gatad2b in this complex. And we've proven by different genetic approaches. Now, to understand biochemically what is going on with this complex and what is the link between Gatad2a, Mbd3, and the rest of the complex, what we did, we took the Gatad2a knockout and Gatad2a wild type cells and IP'd Mbd3 and sent it to mass spectrometry and compared what is the difference between the two. And you can see here that only in the wild type, CHD4, which is the chromatin helicase in the complex, and of course Gatad2a are depleted in the Gatad2a knockout. So we went on to do a little bit what's happening. We showed that Gatad2a is crucial for recruiting the entire helicase complex of the NuRD CHD4. So for example, you can IP flag Mbd3 and induce the Yamanaka factors. And you can see here versus in the knockout versus the wild type, in the knockout, CHD4 is no longer being IP'd when Gatad2a is missing. We did this in also a different assay in the fibroblast, not only in ES cells. Again, if you IP CHD4 and you deplete Gatad2a, you cannot IP Mbd3. So basically, we're looking at an axis of Gatad2a bringing Mbd3 and the rest of the complex. And we also defined the different domains. So in Mbd3, we can delete the MBD domain and we can delete the coiled domain. We go in there and really found that the interaction is actually through the coiled domain of the complex. So you can see here, and again, the IP with the flag, that only in the delta coil domain of Mbd3, you can no longer IP CHD4. And finally, we can rescue this by different mutants. You can see here that in the knockouts of Gatad2a, if you put the wild type Mbd3, you can inhibit the reprogramming. But if you take Mbd3 that lacks this region, you can no longer inhibit this reprogramming. So to make a long story summarized visually, we think of this complex simplistically as containing these parts where we have the Mbd3 bringing the rest of the complex, Gatad2a as the bridge, and this is the helicase CHD4 for the complex. We can abrogate the activity by different ways. We can remove Mbd3 or the coiled region in it, and this will do the job. We can completely knock out Gatad2a. This will do the job. Or we can even develop a peptide. This is again help with the proteomics from Tel Aviv University that have helped us design a peptide, and this peptide that can compete and actually block this activity. So you can see here again, if we add this peptide, we can block the IPing of Mbd3 with the rest of the complex of Gatad2a and CHD4. If we want to go again about what is happening with Gatad2a in ES cells, if we knock it out, you can see that the ES cells also become hyper-naive. So typically you can take wild-type ES cells and change them into primed conditions, and you can see this naive marker goes down. However, Gatad2a knockout, just like the Mbd3 knockout, what I told you, are hyper-naive. They resist becoming primed cells and differentiation. And again, this is also evident by expression of different markers. We are been thinking still at the next level, what is happening with Mbd3 NuRD in development. And this is a figure by our collaborator, Chagai from Tel Aviv University, that we're looking about the evolution of pluripotency genes, and what about the evolution of repressors like the NuRD complex. So you can here you have representation of multicellular organism back to the earliest ones, perhaps T. adherinas. And again, we're looking at Homo sapiens, the humans, and we have values of how much homology to the human sequence there is. And we analyzed actually multiple unicellular organisms. They were all the same, and we just put here Saccharomyces cerevisiae as an example. And if you look, of course, unicellular organisms do not have the OCT4 and SOX2 POW factors. The pluripotency factors started to emerge in the first multicellular organisms, okay? Now, if you looked at HDAC, histone deacetylases, which are found also outside of NuRD, you will find them in unicellular organisms. But if you look at the NuRD-specific components like Mbd3 and Gatad2a, they all three appear in multicellular organisms, and they started to appear in the first multicellular organism. And we're raising perhaps a question here in general about talking about pluripotency or multicellularity. Perhaps that there's, with the emergence of multicellularity, there was the need to make this universal stem cell state, which with these factors emerging, but also alongside with the vector of induction of stem cells that I talked about, these OCT4 and activators, also the factors that terminate pluripotency, like Mbd3 and Gatad2a, also emerge. So we're also starting now to look more, what about these factors doing in different species? I want to highlight, I'm not saying this is the only function of Mbd3 NuRD Gatad2a in stem cells, but I'm saying maybe it started there, and of course they have different roles in adult cells which are very worth looking at. Finally, we're trying to connect the different factors. If we think of how iPS cell reprogramming works, we're realizing that what our model is telling us that we need to reach an activation threshold very quickly. And to reach the threshold, we need different synergistic parameters. I talked on the right, of course, the pluripotency factors, the activators like NANOG and so forth. The signaling is very important, like STAT3 is important. Vitamin C and the oxygen levels, how their effect on the chromatin is very important. But what we added here is this self-repression or auto-repression module, like the Mbd3 NuRD and possibly other complexes that also need to be neutralized. And we are learning currently as well that there are small molecules and pathways that we didn't know what they do before, but it turns out they boost iPS efficiency by inhibiting Mbd3 NuRD. So this is another thing that we're trying to understand. Now, this work on Gatad2a Mbd3 NuRD has been very helpful for us lately to study the iPS cell process. So if you look at conventional iPS reprogramming, where we are dealing with 1% to 10% iPS cells, this makes difficult to work with iPS cells. Let's say our goal is to look at the chronology of success. What happened to the cell that during these 8 days that succeeded in reprogramming? 10% efficiency is problematic, not only because it's hard to sort these cells out, and because we also don't want to look perhaps at the failed cells, but actually when you are, let's say, at day 2, at day 4, you don't know which ones are going to be successful at day 8. And the fact that we have this trick of, doesn't matter if we make Mbd3 hypomorphs or complete knockouts of Gatad2a, we now have a system where about 100% or 99% of the cells succeed in becoming iPS cells. So this is very important for us because now we can play these cells and we know without having to pre-select or predict, we really don't want to make any predictions, that all of the cells successfully convert into iPS cells. And I won't get into the details of this paper we published a couple of months ago, but I just want to show you the power of this platform. And what we did is basically we took these Gatad2a null cells, and what we called it Hanukkah project. It's 8% to 100% day. Every 24 hours, we harvest the cells, and we subjected them to a lot of omics-based approaches about RNA expression, chromatin marks, ataxic, and DNA methylation. And we were able really to learn a lot about the correct sequence of transcriptional activation that is needed to make an adequate iPS cells. And we're interested a lot now because the advantage of the in vitro systems when they are efficient over the in vivo is really now the biochemistry. Because with single cell technologies are not close to really solving issues with biochemistry, gene expression maybe, but with these systems we are now able to look at what is really regulating at the protein level, regulating successful iPS reprogramming. And I think that is going to keep us some of the goals that we will be dealing with in the next couple of years. So with that, I finished. I want to thank collaborators. Some of them I mentioned during the talk, like Gidi Rechavi, Tzachi Chagai, the proteomics at Tel Aviv University, also works here with Ido Amit and Amos Tanai and Yishai Levin. The students, Nofar Moore and Yoach Reis and Shai Geula, that did the work on methylation, continuing with the Mbd3 NuRD complex. I want to thank different funding agencies that supported us, and most importantly, our plasmids are on Agene and protocols are on the website, and we are always looking for talented PhD students and postdocs. Thank you very much, and happy to take questions.
Thank you so much, Jakub, for the great seminar. And please, questions to Professor Hannah. Whoever has, please feel free to turn on the mic. I do have one question related to your last slide. Interestingly, among the battery of techniques that you applied, you also mentioned ribosec, trying to investigate whether there is any component of translational regulation during this process. Usually we think it's mainly or only regulated at the epigenomics transcription. Do you see at all any component of translational regulation in this process?
I must say, actually, yeah, the ribosec we started doing, and we didn't put it in the paper. And as I mentioned, I think this is to me, this is actually one of the most exciting parts. And we just started to delve into that deeply right now. So I don't have any insight to tell you. But I agree. One other thing that we are doing as well is the protein, not only the translation, but also the degradation. Yifat Merbel at the Weizmann developed a very nice technique to look at what's inside the proteasome. And we're very much interested into looking what's in applying this. And I have a new MD PhD student, and this is his project. And this is really one of our top priorities, but we don't know anything yet. I bet that a lot of drama is happening there. I agree with you.
Thank you. Any more questions?
Yes, so I have a question.
Please, Ruti.
So the complex that you have mentioned and the players that are so important early on, do they play similar role in the transition between progenitors to differentiated cells at later stages, or maintaining progenitor pools, interacting with tissue-specific TFs?
Yeah, so I want to give you a lawyer's answer and say yes and no. No, in the sense, I think that not necessarily. So many times we're asked, for example, do you see a similar increase in efficiency in transdifferentiations? You know, for example, there are protocols to make fibroblasts into neurons directly and to ES cells, and we don't see a similar efficiency there. And again, we think this is because, as I mentioned to you, ES cells and iPSs are the only cells that can really tolerate complete loss of these repressors. And versus, for example, neurons, they also stop proliferating with it. Having said that, there's very, very little work on this complex and these components in adult cells. The very few paper, there are 2 in numbers, And yes, so there's a Nature paper that showed Mbd3 knockout cells block formation in the intestine of the maturation of the progenitors towards the colony and can affect tumors. So that can suggest that definitely they're playing a role in that pathway. Now we're trying to make the conditional, we've been a little bit unlucky, but we're hopefully close to make the flox for Gatad2a. And this is again one of our goals to work to look at this. So and I bet there is a lot of in it, yeah.
And this MDB3 hypomorph, is it in the mutation there? Is it in the cold domain? How is it related? Do you know if it's?
It's interesting. No, so they removed exon 1 and exon 2 that makes an out of frame, these mice. And there are two mice that were made from this, and they behave a little bit differently. So that's why a little bit also so you would expect usually the flux minus, you will expect 50% protein level. And so one of the mutants is like that, and the flux minus are viable completely. Another mutant, the flux minus, which I showed you that it's 20%, and the mice are not viable. So you can make ES cells, you can make different tissues, but an entire mouse is not viable. So that's why a little bit we basically took advantage of this, unfortunate why this mutant is behaving like that. But we now understand we have more control with the Gatad2a. But nobody has made mutations about the coil concept. So exactly, we're trying to make an Mbd3 mutant that lacks the coil domain and ask basically, does it behave more like wild type? Maybe the mice are viable, but we can get the effect on iPS cells. So we don't know yet. That's something also we're trying to do.
Thank you.
Thank you. Any more questions to Professor Hannah? Okay, if not, many thanks again for the great seminar. And thanks for accepting this challenge. I think it was great. Thank you. So all the best to you all. Thank you.
Thank you.
Bye-bye.
Thank you.